Hi there, my name is Nick Bernstein. I'm here to talk to you today about a tool for doublet identification in single-cell RNA-seq via semi-supervised deep learning. The tool is called Solo, and it was developed by me and my colleagues at Calico Labs. So doublets are a problem that are currently still inherent to single-cell RNA-seq. They come about because after you've dissociated your cells, or if you've taken your cells from cell lines, you have to go through the process of encapsulation. During encapsulation, depending on how many cells you load, you will get doublets. These doublets can be particularly problematic when they come from two distinct cell types. So on this plot over here, we have a low-dimensional embedding of some mock data in which we have two cell types, and we have doublets of those cell types. Doublets with two different cell types, which we call heterotypic doublets, can be particularly problematic because they create these non-existent transcriptional states, which can lead to improper biological inferences about new cell types or new cell states. The way Solo identifies doublets in single-cell RNA-seq experiments is by first creating a low-dimensional embedding of our data. We use the tool SCVI, which is a variational autoencoder for single-cell RNA-seq, to do so. On the right here, we have some example mock data, which is our observed data with a cell type A, some doublets that we've observed, and a cell type B. But we don't know a priori that these cells are doublets. And if we wish to train a classifier to identify these, we need to create some doublets. So for the purposes of Solo, we'll treat all of our observed data, which we see here, as singlets for our classifier. So the next step is to generate a positive class of doublets. The way we simulate doublets is through the simple operation of addition. We randomly sample pairs of cells, add their gene expression vectors together, and get a new gene expression vector, which we consider a simulated doublet. And this ends up being our positive class for our classifier. Once we have our simulated doublets and our observed data, which we treat as singlets, we embed the gene expression vectors X into the latent space Z using SCVI, and from this latent space we train a classifier. The classifier we've chosen to use is a simple multi-layer perceptron, and we train with early stopping, dropout, stochastic gradient descent, the typical bells and whistles of deep learning today. And we find that this works quite well. We validate Solo by using the ground truth labels from experimental doublet identification approaches. There are multiple tools for doing so. One is called Multiseek, which uses natural genetic variation. In this case, it was done on unstimulated peripheral blood mononuclear cells, PBMCs, from different patients of different genetic backgrounds to identify doublets experimentally. We find that Solo has better agreement with this demuxel method than other computational doublet detection methods, as seen here in terms of average precision and on this precision recall curve. We also find that Solo has better agreement with cell hashing. In this case, 4 cell lines were hashed together to identify doublets, and Solo again has greater agreement with these doublet calls than other computational doublet detection methods. The next question we wanted to ask ourselves was how well would doublet detection work in a complex solid tissue? To do this, my collaborators took a couple of mouse kidneys, performed tissue dissociation, then performed Multiseek by breaking up the sample into 8 different aliquots, and then performed pool staining and FACS to remove dead debris and enrich for live cells. We found, again, in this context, that Solo outperforms other computational methods in its agreement with Multiseek. Here we have the 2 different mouse kidneys that were run and the agreement of Solo with the Multiseek calls. Lastly, we wish to see how well Solo could complement current experimental approaches. Many experimental approaches currently you get for free. In the case of demuxlet, you might already have some natural genetic variation in your pooled samples for your single-cell RNA-seq experiment. So we wanted to ask, how well could solo and demuxlet be combined to have better agreement with, for instance, cell hashing? To do this, we took an experiment from the original cell hashing paper in which they took PBMCs from genetically diverse individuals and hashed each one of those individuals with its own unique hash. So what we can do is see what the agreement is between demuxlet and cell hashing, which we see here is quite good. And we can also see how well solo from the transcriptional data can identify doublets, which it can do as well, but not as well as demuxlet. But what works best is when we average the scores from solo and demuxlet together to get the best agreement with the cell hashing labels. To us, this gives us a strong indication that solo can be used in combination with experimental approaches for identifying doublets. Thank you so much for listening, and I hope you find solo useful in your future research.